Welcome back to the continuation session. And we were talking about the functional characterization of a gene which we fished out from self-differentiating system. As you recall, the objective was to understand the molecular basis of cell differentiation using in vitro cell differentiation system, and we ended up isolating a gene whose sequence resembled with that of or showed identity with wall-associated kinases, which belongs to a family in rice. There are 125 members of this, and before we started working on to functionally validating this, none of the gene from rice was validated for its function, except that in Arabidopsis, two of its members were shown to be having a role in pathogenesis or pathogenesis-related responses, and also one of them had a role in development, like controlling cell elongation, basically. So, and this gene showed differential expression in an organ-specific manner. It expressed itself only in roots and panicle, and of course, it is differentially upregulated by hormones and protein, and downregulated by stress signals like salinity, abscisic acid, salicylic acid, etc. So this then encouraged us to proceed further to functionally validate this by using silencing, gene silencing approach. And for this, what was done is this, that you have this vector, what we call PWAK RNAi 6.72 kilobase pair. This is the size of the gene which has a promoter, 35S promoter, and OCST terminator. And now for generating silencing cassette, we have to have very much gene-specific region identified. And in this case, we identified 400 base pairs which were unique to the gene, which was cloned, sense and in antisense orientation, which was having an intervening sequence. It is the PDK intron. PDK is phosphodide orthokinase intron from maize, actually. So this is done in order to generate a hairpin-like structure, which then use the RISC process and try to chop off the transcripts made in the cell, And this silencing is specific because of these sequences here, which are from gene in general, and so whatever gene these sequences you are taking, it is going to act upon the transcript of that gene very specifically, So, so the transgenic plants were produced using gene gun method, which I had been talking all along. It tries, and five lines were identified: 1, 2, 3, 4, 5, and this is the control. And again, to check whether these transgenic lines, which were basically identified based on their survival on antibiotic resistance, or they could grow on a medium containing antibiotics like hydromycin phosphotransferase. So, if you see here, the genomic DNA was taken uh, from the transgenic lines, and one from control, which is untransformed plant, run it on a gel, and then very specifically, this plot was probed using this region, that is the PTK from maize and from, now this sequence is not in rice, and therefore, if this cassette is present in the plants, it's going to pick up very specifically this cassette actually. So this is what is happening when you use this as a probe. You can see these transgenic lines showing hybridization to this probe, indicating that the gene has gone in. Further, one has to find out whether this gene is expressing and doing its job. And what is its job? The silencing. So when you look for the presence of transcripts, like RNA is isolated, extracted, run it on a gel, and then probed with the gene probe cDNA with which we started. So you can see in control plant, you can see the presence of transcripts by way of the probe hybridizing here. But if you see in transgenic lines, except this, which showed little bit of expression, none of the 2, 3, 4, 5, these lines show no expression actually. So it means the cassette is working and degrading or the transcript made by the original gene of the cell. This is one indication that the transcript is being destroyed by or being silenced, in other words, what we say. And another thing is that if that is true, then you should be able to see these chopped off transcripts, which generates 21 nucleotide pieces of the entire transcript. So that was checked here, 21 nucleotide. So in control, we don't see any, and in all the transgenic lines, you can see the accumulation of these nucleotides, which indicates that the bigger transcript got chopped off into 21 nucleotide pieces, long pieces. So indicating thereby that the gene is being silenced and there is no transcript in the system. And when there is no transcript, there is no protein. So it means we had transgenic lines where this gene has been silenced. Okay, now if that is true, then what is happening? So in the T0 plant, T0 means the tissue culture regenerated plants were looked at, and you can see, if you see, this is the control plant inflorescence, and this is the transgenic line. So if you see, this is the flag leaf, we call it, which covers the inflorescence. This is the flag leaf, normal, and in the RNA silenced plants, it is very, very reduced, So one difference which one could see visually is this, the size of the flag leaf, and also if you look at the inflorescence, that is also reduced. Similarly, if you look at here, this is the size of panicle. This is the panicle I have been referring to. It is very, very short. They have come in the transgenic plants where the gene is silenced as compared to the normal. And this is the leaf size, and details of the panicle here. This is the normal panicle, and this is the panicle from the silence. So, and also if you look at the phenotype of anthers. This is the normal scenario where the anthers dehisce to release the pollens, which fertilize and leads to the uh, the the fertilization and the seed development. But in the transgenic lines where the gene is silenced, you know, the entherdiscence is very less, or they don't dehisce, and when they don't dehisce, they don't release pollen. So there were visual difference which could be seen in the during development. You know, and then next question which we looked at, these pollens here and here, when they released, what is the viability? So viability of these pollens was checked, and it is very interesting to know that the pollens were that 60% of the pollens were not viable in the silenced plants as compared to control. So not only this silencing is affecting the size of the panicle and the behavior of anthers, but also it is affecting the viability of the pollens. In the silenced plants, 60% of the pollens are non-viable. And this is checked by a simple pollen starch test is done, which gives you shows you the viability of the cells, of the pollens. Now, when we looked at the uh, in the mature plants, what is happening if you compare different parameters, you can see height of the plant. Normal plant is about 74, average height is 74 centimeters, and the silenced had 54. So it shows dwarfism. Actually, the transgenic plants were dwarf or shorter in size. And then number of panicles per plant was more in the transgenic lines as compared to control. And the size, but if you look at the size is reduced. Here it is 18 odd centimeter to 11 centimeters. And number of spikelets per panicle, this is interesting. It is 100 here, and it is uh, decreased by almost 60%. So you have only around 40 to 45 spikelets per panicle as compared to control. And very interestingly, if you look at the number of mature seeds per plant, in normal plant it produces about 500 to 600 seeds per plant. In the silenced, we could see only 3 to 4 seeds per plant. So visually, it was affecting the plant productivity. Means the silencing of this gene has affected the overall growth of the plant, and which resulted into the reduction in the number of seeds per plant, which is 3 to 4 plants. 3 to 4 seeds per plant as compared to this. So it's very interesting observation here that the silencing of this gene is affecting morphological traits like the shoot lengths, sizes, leaves to flag leaf sizes, panicle size, the number of panicles per plant, and finally affecting the number of seeds per plants. Okay, so whatever seeds were collected here, and of course we had seeds from control, the further analysis was done in the T1 generation. So if you look at this is very interesting observation. So if you germinate the seeds, and this is what one can see. These are the control seeds germinating normally. They look healthy. And if you look at the seeds from silenced plants where the walk gene is silenced, you can see visual differences here. These are the different 5 lines which were picked up. You know, they are showing very little root growth here and very less shoot growth here as compared to control. If you see here, so it affected the structured root growth. You know, if you see. If you see here, you know, these roots have characteristic root hairs. So this is the normal, you know, root with root hairs, but you know there is no root hair in the seeds from the silenced plant. So it means the silencing of this gene is affecting root growth as well as the root hair formation in the root, and also, if you see, um, if you see in the next slide, that is the lateral root formation. This is the normal lateral root emerging from the primary root. There is no lateral root formation in this silence. So, uh, so if you look at these observations, that the silencing affected normal root growth. Okay, uh, if you see here in the T1 generation, when the similar studies were made with the inflorescence, you can see panicle is small sized, you know, and no seed set in this. You know, if you in the T0 plants, we could at least collect 3 to 4 seeds per plant, but in T1 generation, they only showed these kind of inflorescence, but there was no seed set here. Okay, so in other words, the plants were completely sterile. You know, so and also if you look at the root, you know, this is the normal plant root growth after 35 to 40 days. Plants when they were harvested, you can see this is the normal root, but in the silenced plant, the root growth is completely uh, inhibited. You know. So root biomass was completely inhibited. You can see here, this is the normal, and these are the silenced plants. So to conclude this part, uh, we had this RNAi mediated silencing of this gene resulted into uh, impaired root development and sterility due to anther indehiscence. The plants were sterile because anthers were not able to dehisce. Okay, so the details of this work is in this paper. I think I have put it on in the Google Class. If not, I will do that. So you can go through the finer details of this paper here. But then to conclude, um, what I have tried to do is to give you an idea that how one can very systematically look at the various steps involved in functionally characterizing, isolating a gene and then functionally characterizing. In this case, this is the gene which was found to be involved in root development and also in the reproduction in terms of affecting the root, affecting the pollen sterility or pollen indehiscence. Sterility is because of pollen indehiscence. You know. The other aspect of this is instead of silencing, if you overproduce this gene or overexpress this gene, then what will happen? You know, you will have more healthier plants. You know, there will be more root biomass, and also there will be very effective dehiscence of the pollen, and the productivity can be increased. Although we have not done this, by overexpressing this gene, what happens? But it can be concluded that if you overexpress, then it can be again a candidate gene for improving the plant productivity in the